Okay, in this part, I'm going to talk about flow cytometry basic principles and some of the steps that everyone needs to follow. Sample collection. In our study, since we wanted to define normal range for patients, we took blood sample from healthy volunteer people in different age groups. By the way, this one doesn't seem so volunteer. Sample preparation. In the study we've done, we wanted to target some markers on the surface of white blood cells called CD markers. Unbalanced expression of these markers might show different diseases. If we want to detect them, we need some specific antibodies. Specific antibodies are designed to identify specific CD markers. They can bind to the marker and they can visualize the presence of them directly and indirectly. Here I'm going to talk about direct method. To be able to see whether antibody identified marker or not, they are conjugated with a fluorescent substance called fluorophore. In direct method, the fluorophore is attached to the antibody directly. Indirect conjugation. In indirect method, primary antibody targets the antigen, secondary antibody targets the primary antibody, and fluorophore is attached to the secondary antibody. Flow cytometry and data acquisition. In this level, the technician will put the tube, including the sample, in the flow cytometer and the sample will be sucked up by the device. Inside the flow cytometer, sample stream enters the flow cell which contains sheath fluid. Due to the pressure difference between sample stream and sheath fluid, cells will be concentrated in the middle, and this phenomenon allows the cells to pass through the laser beam one by one. Cells pass through the laser beam one by one at interrogation point, and the laser beams scatter to different directions and hit the detector. With detecting the scattered beams, cells can be analyzed. There are different types of scattering. When beam scatters along the path of the laser, it's called forward scatter, which corresponds to the size of the cells. Another type of scattering is side scattering. When the beam hits the granules inside the cells, as they are dense, it bends and it's called side scatter. It shows the granularity of the cell. Finally, when laser beam hits the fluorophores which are attached to the antibodies and markers, it emits fluorescence, and by detecting this fluorescence and analyzing it, we can reach to the chemical properties of the cells. We usually use different fluorophores, which means they have different wavelengths and emit fluorescence with different colors. In this device, there are dichroic mirror, long pass, short pass, and band pass filters. Dichroic mirror and filters allow fluorescence with specific wavelengths to pass through them and reach the detectors, while other wavelengths will be reflected. Beams will be collected by the detectors. The main part of the detector is photomultiplier tube, which converts the detected beams to the signals, amplify them, and send them to the computer. Computer converts them to the data. Data visualization and dot plot. Dot plot is a popular way for flow cytometry data visualization, and here you can see forward scatter and side scatter chart in addition to the chart that shows the percentage of the cells that express the targeted CD marker. Let's focus on forward scatter and side scatter chart first. As I mentioned, forward scatter corresponds to the size of the cell and side scatter shows granularity. For this reason, the more we go to the right, the dots represent the bigger cells, and the more we go up, the dots represent more granularity, so the cells are more complex. And if you know white blood cells, you will understand why granulocytes, monocytes, and lymphocytes are placed in the picture like that. And here you can see the distribution and the percentage of the same cells which are expressing some specific CD markers on their surface. And finally, I hope this animation provides you with some basic information about flow cytometry. I tried not to go into details. For this reason, sorry if I missed some parts. Thank you.